Hello, everyone. Today, I would like to discuss how gene expression is regulated. For this part, we only have very limited information from the textbook, so please follow my lecture and slides for your study. Gene expression is a process of making proteins from genes. As we learned, every single cell in our body contains the same genomic DNA, but these genes are not turned on at the same time. Actually, only about 5 to 10% of genes are expressed in any given cells. For example, the brain cells may make proteins for producing neuron transmitters, but they do not make proteins to digest food. The process of turning genes on or off, or making more or less proteins, is called gene regulation. Based on the phase of gene expression, gene regulation can be controlled at 4 different stages. Transcriptional control that regulates the efficiency of transcription, or RNA processing control that regulates the process of pre-mRNA to mature mRNA, or RNA transport control that regulates the delivery of mRNA to cytoplasm, and the translational control that regulates peptide synthesis on ribosome. In eukaryotic cells, the start of transcription is one of the most complicated parts of gene regulation and also most studied. We will focus on this topic today. I showed this slide before. To refresh your memory, a gene is composed of a gene body and gene regulatory regions. A gene regulatory region is a segment of DNA that is capable of increasing or decreasing the expression of specific genes. Okay, showing here, enhancer and promoter, they both are regulatory regions. First, let's see what is the promoter. Okay, promoter is a DNA region of 100 to 1,000 base pairs that can initiate the gene transcription. We mentioned that promoter is right in the upstream of the gene and coordinates with enhancers to regulate transcription. So if I ask you a question, where does transcription initiate in a gene? You should know the answer by now, okay? So what is so special about promoters? One feature of promoter is that it contains a specific DNA sequence called a Tata box, okay? It is located about 25 to 35 base pairs upstream of the transcription starting site. Transcription starting site showing here as an arrow here, and has a consensus sequence that contains TATA. So that's why a Tata box coming from. Interestingly, a protein names, this TBP stands for Tata box binding protein, can bind to the Tata box. It can also bend the DNA and separate DNA double strand. Okay, so TBP also brings in more transcription-related proteins and initiate the formation of the RNA polymerase transcription complex and setting it ready for a gene transcription. At this point, a very low level of transcription could occur. We call this type of transcription as basal level of transcription or just a basal transcription. Since TBP can bind to DNA and is transcription related, it is considered as a transcription factor. However, it is a general transcription factor as every single cell has TBP and it is needed for all gene expression, all right? So here you can consider general transcription factor as a constitutive transcription factor, all right? So in contrast to general transcription factor, there are regulatory transcription factors which are only expressed under certain conditions. We will get into that in a few minutes. Then how does promoter and enhancer coordinate to regulate transcription? As we learned, an enhancer can be located far away from its regulated gene. Sometimes enhancer is one mega base pair, meaning like 10 to the 6, 10 to the power of 6 base pair away from its regulated gene. Okay, so in most cases, an enhancer is located upstream of its regulated gene, but sometimes it can be downstream or even in the intron region of its regulated gene, right? So enhancer is a short DNA region. It composes about 50 to 1,500 base pair that can be bound by transcription factors to activate transcription. An enhancer does not work by itself, as the interplay between enhancer and promoter is essential for transcriptional activation. Although there are different hypotheses on the precise function of enhancers on transcription, it is generally accepted that enhancer actually forms this loop to the promoter region and activate the transcription. We call this as a looping theory. Indeed, these days, scientists can detect these chromatin interactions through genomic approaches, such as like Hi-C technology, providing direct evidence for looping theory. I just mentioned that transcription factors combine to enhancers region, right? So interestingly, majority of transcription factors that bind to enhancers are regulatory transcription factors, but not the general transcription factors such as TBP. So this is one big difference between enhancers and promoters, okay? So then one question is, does enhancer contain Tata box? No, okay? Then how do transcription factors bind to enhancers? This is because transcription factors can read a special short DNA sequence and bind to this piece of DNA. This sequence is named as transcription factor response element. For instance, Tata box is the response element for TBP protein, right? So different transcription factors recognize different response elements. So scientists that have studied many binding sites for many different transcription factors, and there are softwares that can predict the transcription factor response element simply based on a given DNA sequences, all right? So showing here is an example. This is a consensus sequence of mouse of transcription called a transcription factor called the FOXO1 response element. Okay, FOXO1, of course, is a transcription factor, and it binds to the sequence of this G, C, T, G, 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 and either G or C in the position 7 locus, and then T, T, T. All right. If you find a match sequence on gene enhancer region, there is a good chance that this gene can be activated by FOXO1. So now let's watch a short video on transcriptional initiation.
For RNA polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short TATA sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors, called coactivators, link the basal factors with a third set of transcription factor proteins called activators. Activators are regulatory proteins that bind to sequences on DNA called enhancers. Enhancers are located at sites that are distant from the promoter. The interaction of activator proteins with transcription factor subunits increases the rate of transcription. Many enhancers scattered around the chromosome can bind different activators, which provide a variety of responses to various signals.
This picture is similar to what has been shown in the video, except that the colors are different. The activator in the video refers to a transcription factor. From this picture, you can see that one gene could have multiple enhancers. Showing this picture, there are 3 enhancers. Meanwhile, each enhancer has multiple transcription factor response elements, meaning more than 1 transcription factors can bind to one single enhancer. Although in this picture, only 1 transcription factor binds to one enhancer. Okay, so the more proteins bind to each other can make the loop structure much stronger, or you can say more stable, so allowing a more efficient transcriptional initiation. This is the reason why we think transcription factors are the driver of transcription. Based on what we have learned, now you can see that one important feature of transcription factor is that it can bind its response element, which is a DNA, right? So this is the reason why all transcription factors contain DNA binding domain, or abbreviated as DBD. Transcription factor is the single largest family of human proteins, and approximately 10% of genes in our genome code for transcription factors. But do all transcription factors express in every single cell? No. General transcription factors, such as TBP, may be expressed in all cells, but for majority of transcription factors, their expression is conditional. Okay, I'll give you some examples next. Scientists have tried to classify transcription factors. Here shows the classification of transcription factors by their function. So this is a functional classification. You may notice that here it says positive acting transcription factors, which implies there are negative acting transcription factors, meaning some transcription factors repress transcription rather than activating it. However, since there are much fewer studies focusing on repression function of transcription factors, in this class, we only focus on transcriptional activation driven by transcription factors. But I want you be aware of that. There are some exceptionals. So the transcription factors can be generally characterized into 2 families. One is they constitutively active. We learned the TBP, which should belong to this family, okay? Another family is called transcriptionally active, conditionally active, meaning they can only be activated in certain tissues or when receiving certain signals, all right? For instance, the MyoD protein drives the muscle cell differentiation, so it is only expressed in muscle cells. And the other one is PIT1. This is a transcription factor for growth hormone and is a pituitary specific transcription factor. Okay, so you don't have to remember these specific names, but you need to understand the meaning of each category, all right? And the signal-dependent transcription factors can further be classified into these several families, depending on different activating signals. Right? So the steroid receptor superfamily is a group of transcription factors that can be activated by hormones. Also, there are other groups such as being activated by internal signals inside the cells, or activated by signaling from cell surface receptor stimulation. We will not get into details on those. Instead, I would like to spend more time further introducing the steroid receptor family. The steroid receptor family belongs to the steroid receptor superfamily, and the 5 transcription factors are included, okay? AR is androgen receptor, ER is estrogen receptor, and PR is progesterone receptor, GR is glucocorticoid receptor, and MR is mineralocorticoid receptor. You should be familiar with some of those receptors. AR, ER, and PR are also called sex steroid receptors. These proteins interact with sex hormone androgens, estrogens, and progesterones, respectively. The picture here shows you how steroid receptor family of proteins work as transcription factors. This is a part of the cell showing the cytoplasm and then the nucleus, okay? So when steroid hormone here exists in our bloodstream, it diffuses into the cell. In cytoplasm, the hormone has chance to encounter the steroid receptor protein. And then since a steroid hormone has a high affinity with this receptor, it binds to the steroid receptor and forming this hormone receptor complex, all right? So leading to the nuclear translocation of the steroid receptor. Remember, these receptors are transcription factors. So once they enter nucleus, they can bind to their response elements, help to start a transcription and produce proteins that may involve in the cellular responses, right? Let me show you an example to see how hormone-induced transcription is related to our everyday life. And now we can take this action to, develop all kinds of drugs. So this picture shows the changes of several hormones during pregnancy. You may notice that the levels of estrogen and the progesterone, they keep increasing with pregnancy. Progesterone is also called as hormone of pregnancy, as it has many roles relating to the development of the fetus. For example, progesterone prepares endometrium to the best condition for implantation, okay? Progesterone can also decrease contractions of uterine smooth muscle, helping the uterus to increase in size and preventing preterm labor. Then estrogen is also extremely important. A woman will produce more estrogen during one pregnancy than throughout her entire life when not pregnant. Estrogen helps to form additional blood vessels, which can accommodate the nutrient support of developing fetus. It also helps to develop milk ducts for breastfeeding. Okay, all these hormonal effects are carried out by steroid receptors. Here, the PR and ER. Okay. Also, including this picture, you may notice the other hormone called the human chorionic gonadotropin. Okay, so this is also abbreviated as HCG. So it is peaked during early pregnancy. HCG is produced by the trophoblast cells and promote the corpus luteum to secrete the progesterone during the first trimester, okay? HCG is now used as a marker for early pregnancy. So now it appears that these hormones are very important for fetus development. What would happen if the signaling pathways are blocked? Here I give you an example, okay? This compound RU486, also called mifepristone, is an abortion medicine, okay? If you compare the chemical structure of RU486 and the progesterone, you can tell that highly similar. So therefore, RU486 works as a competitive molecule for binding with PR. When 486 binds PR, it occupies the binding pocket, and the progesterone can no longer bind with PR and activates downstream genes. So as a result, the progesterone signaling pathway is completely abolished. Okay, in patients, RU486 breaks uterus lining and causes bleeding. And then RU486 also opens the cervix, leading to contractions that help dislodge and expel the embryo. So then the pregnancy is ended. All right. So RU486 is very effective on abortion. When combined with the misoprostol, it's more than 97% effective in ending early pregnancy. So this drug is approved for use in the United States in 2000. Okay, based on what we have introduced, I would like you to remember several definitions. All right, so first is ligand, which refers to a molecule that binds to another larger molecule, like estrogen, progesterone, testosterone, RU486 are all ligands because they could bind to ER, PR, AR, and PR again, respectively. All right. Depending on different functional consequences, ligands can be further classified into agonist or antagonists. So agonist is a substance that fully activates the receptor protein that it binds to, like estrogen, progesterone, testosterone, and those are the examples of agonists. But in contrast, antagonist refers to a substance that binds to receptor protein but does not activate or even block the activity of other agonists. A good example is called, we learned this RU486. Okay, now let's take a look at the androgen receptor or AR signaling pathway. During the lecture of sex determination, we learned that testosterone is important for development of male reproductive organs. At the molecular level, this process is mediated by AR. All right, so in our body, the testosterone can be converted into a molecule called dihydrotestosterone, also known as DHT. Okay, T and DHT both are androgens, and DHT is more potent than T when activating the androgen receptor. Okay, so the AR protein has 3 major domains based on its function, named this N-terminal domain and the DNA binding domain in the middle, and the ligand binding domain on the C-terminals. We learned that all transcription factors have DNA binding domain. Is that correct? Yeah. So this domain is, the DBD is responsible for AR to bind to DNA or chromatin by recognizing its response element, right? Also, as it names, the ligand binding domain is where the ligand bind. So T and DHT were binding to somewhere in this domain. Interesting, the binding of T and DHT can change the structure of the AR protein and further recruit transcriptional co-activators to initiate the transcription. Under this condition, you know that both T and DHT are agonists. Let's see how AR signaling works in a cell. Okay, when DHT enters the cell, especially binding to AR protein, the interaction changes the AR protein structure and directs its nuclear entry. All right. In nucleus, the activated AR binds to ARE in the chromatin and activates the gene transcription. So here, the genes regulated by AR are called AR target genes. So how many AR target genes are there in a cell? Take a guess. Is it 1 or 2 or more? Actually, there are probably hundreds or more AR target genes. Okay, so you can imagine any gene with ARE and have an open chromatin may have a chance to be activated, right? So they function together to promote the cell proliferation or development of male reproductive organs, depending in which cells and when the signaling pathway is induced. So this is a target gene. So in prostate cancer, AR signaling is essential for prostate cancer cells to grow or to proliferate. Therefore, scientists developed ways to inhibit this signaling pathway. There is a compound named enzalutamide combined to AR and inactivate AR transcription factor function, leading to cancer cell death. So it has been widely used in clinics to treat prostate cancer. And then this enzalutamide actually can bind with the AR competitive with the DHT binding. So based on those this description, you should know that enzalutamide is an AR antagonist. So we also have learned androgen insensitivity and showed you, I showed you this slide in lecture 9. By then, we did not mention what kind of mutations on AR could lead to androgen insensitivity. Okay, there's mutations happening in this gene. So this figure actually summarizes the mutations found in individuals with androgen insensitivity syndrome, also called AIS. Okay, AR is a protein with 919 amino acids, showing on the top here, and it contains 8 exons labeled here. And this is the AR gene, also contains, you can see 8 exons. While these mutations are disrupted throughout the entire gene, you can see all these mutations, and also in the exon, intron somewhere over here, for instance, and some are mutations here. But you can see this majority of those mutations are concentrated in several regions in AR protein. Okay, if you located those to the different domain of AR, you can find out a lot of them are located in the DBD, DNA binding domain, and LBD, ligand binding domain. Okay, you can imagine that DBD, if they have mutations, AR protein may not be able to find its response element on its target genes, and consequently, its target genes are not expressed, right? So, but then if LBD has mutations, the mutated AR may not be able to bind ligands or cannot bind the transcriptional co-activators. Therefore, the target genes cannot be activated either. Okay, so these are the molecular mechanisms of how AR mutations contribute to androgen insensitivity syndrome. Now let's spend a few minutes discussing the transcriptional co-activators. They are the group of proteins shown this in blue. You can see that these co-activators can physically interact with transcription factors, but not DNA or chromatin. So a co-activator is a type of protein that can bind to transcription factor to increase the rate of gene transcription. Co-activator proteins have several features. First, they cannot bind to DNA directly. Instead, they have to interact with transcription factors and are recruited to chromatin. So this is the reason why they are called co-activators, not activators, right? So many co-activators are enzymes that modify chromatin, and many of them function together as a protein complex, similar to like a teamwork. In addition, the function of co-activators is to facilitate transcription. They are co-activators for transcriptional initiation, also co-activators for transcriptional elongation. But how co-activators facilitate transcription? What do co-activators do? This figure shows you several groups of co-activator complexes. First of all, in the bottom, this is the DNA arranged in the format of nucleosomes, right? So the labeled specific transcription factor are hormone-bound nuclear receptors. Nuclear receptors are a bigger family of transcription factors, and all steroid receptors are nuclear receptors, okay? Anyway, they are transcription factors. Based on this information, you should be able to tell that this is actually the enhancer region, right? So the binding of transcription factors further recruit these co-activator complexes through the direct interaction between transcription factors and the co-activators. Okay, so these co-activator complexes have different functions. Okay, first of all, the this is BRG-BRM complex is an ATP-dependent chromatin remodeling complex, meaning the protein in this complex can utilize the ATP or energy to remodel, or you can say change the chromatin organization, all right? So the SRC co-activator complexes, they contain enzymes that can modify histone proteins. The modifications include acylation and methylation, okay? And we will discuss this in detail in our next lecture. And similarly, the ASH2, ASH2 complex can also methylate the histone proteins, but on different amino acid residues on histones, okay? The co-activators complex also include this mediator complex. And then the mediator complex can interact with both the transcription factors as well as the RNA polymerase II complex and facilitate the looping, the enhancer and promoter looping, right, between the enhancer and promoter regions. So let's further take a look at the chromatin remodeling, what this chromatin remodeling really means. Remember, the default status of genome is a repressive status. We learned that the heterochromatin has highly compacted chromatin with very few genes. But even in the euchromatin region, that is enriched with genes and relatively loosely packed, the nucleosome structure is not always accessible for transcription factors. Showing here, like they may embed it inside the nucleosome structure. So the chromatin remodeling is the rearrangement of chromatin from a condensed state to a transcriptionally accessible state, allowing transcription factors or other DNA binding proteins to easily access DNA and control gene expression. In this figure, the remodeler activity actually ejects or kicks off the two nucleosomes in this promoter region, and then create a nucleosome-free region for binding of other transcription-related proteins, including a transcription factor shown here and the co-activator shown here. But then in some other times, ATP remodeling can also reposition nucleosomes by breaking the interaction between DNA and the histone proteins. So it is also often referred as nucleosome sliding, okay, along this DNA strand. So the ultimate purpose of chromatin remodeling is to make DNA sequences more accessible to transcription factors. Okay, now I would like to show you how to read gene information in genome browsers. A genome browser is a graphic interface for display of information from a biological database for genomic data, okay? Genome browsers enable researchers to visualize and browse entire genome with gene prediction and structure, proteins, expression regulation, comparative analysis, so on and so forth. A large number of genome browsers are available. Many of them are free and with database accessible online. They all look very similar, okay? Here I'm using this Integrative Genomics Viewer, or IGV, as an example to introduce some basic information, okay? IGV is a software that you can download and run in a local computer. This shows a screenshot of this IGV window, okay? Based on what we have learned, you should now be able to understand all the information in this picture. So I will go through this one by one with you. Okay, first of all, in this window, it shows the human HG19. This indicates this is a human genome showing in this genomic viewer. And this is a version HG19. With the advancement of genomic sequencing technology, the human genomic information becomes more and more accurate. Therefore, the version of genomic information has been updated periodically, all right? So the chromosome 8 here means chromosome 8. So, and then here indicates the location of chromosome 8, which is showing from here to here. This is 12 kb in this region, all right? But if you look into the chromosome locus on the cytogenetic bands, showing this red color here, so you can tell that the region is on the chromosome, it's a long arm of chromosome 8, and it's located in the Q24.21, showing here. So that's the region, the red bar over here shows this region, 12 kb region as well, okay? In this region, it has two genes. One is MYC gene, another one is called the CASC11 gene over here, okay? And the CASC11 should have at least two alternative splicing forms showing here, because one you can see a complete splicer form has two exons, but the other one is just showing partially, but then you have at least two different splicing form. But MYC showing here only one form, and then you can see the whole gene of MYC. The arrow points out in the direction of gene. For instance, the MYC gene showing here is from 5' to 3', but transcription starts always at 5' and goes to the 3' direction. All right, so the arrow shows the direction. So you can tell where is the 5' of a MYC. It's on the left, okay? MYC gene has 3 exons, showing those bars over here, 1, exon 2, and exon 3. And then you may notice that in the exon 1, this majority of the regions showing are more narrower than the other regions. So you will tell that, what is this region tell? It's 5' untranslated region, 5' LTR, okay? And as 5' UTR. And then based on this, you can also tell that in exon 3, so this region is the translated region, but in over here, the more narrow the fragment here is 3', because on the end it's a 3'. 3' untranslated region, 3' UTR, okay? And then based on this, you can also tell that transcription actually starting from here and end here. But translation start from here, but then ends here. So then if I'm asking you where is the translational starting site, like AUG or ATG, where they're located, and then you will be able to tell me that's somewhere over here, okay? So make sure you can read all this information because it will be included in the exam, all right? So you can also estimate, you know, how long, for instance, is the MYC gene. If you know this is 12 kb, and then you can estimate probably this is I would say 5 kb. Right? So that's the length of the MYC gene. So all this information you will be able to tell just by looking all this the graph showing here. Okay, lastly, let's go over some practice questions. Okay, so let's see the first one. mRNA, this is true or false? mRNA is made of introns that have been spliced together. Is this true or false? So you may say, oh, I know this one. This is false because it should be exons, not introns, right? Okay, the second one, the codon encoding the amino acid methionine functions as a start codon where it occurs at the beginning of a gene. Is this true or false? The answer is true because the AUG or ATG encodes methionine and it is the start codon for translation. So that's correct. Okay, the third one. The set of proteins in a particular cell type is always equal to the number of genes in the genome. Is this true or false? So last lecture we'll talk about that. So the answer is false. So the set of protein is always larger than the number of genes. Why is that? We learned that alternative splicing and the post-translation modification are all contribute to it, right? Okay, so multiple choice questions. The first one, peptide bonds are formed between what? So this should be pretty obvious. So the answer is D, from amino group and a carboxyl group. Okay, so this is very straightforward. Second, one gene is able to encode information for different form of protein via what? So the answer is A. We never talk about the primary splicing or secondary splicing. So B and C are not correct, right? And then E apparently is wrong too, right? tRNA mutations. But then only D is a little bit confusing. But first, mutations are abnormal, and this question is not asking a disease condition. So it does not really fit. Second, when we talk about mutations, we usually refer to mutations at DNA level and protein level. We rarely mention the mutations at the mRNA level. So only A is the right answer. It's so much better than D, okay? So see this one. The tertiary structure of our protein is what? You can take a look. So B is the correct answer. So A refers to the primary structure because amino acid in a polypeptide chain, right? C is not an accurate description. Tertiary structure is not limited to a protein region, and sometimes it needs a chaperone to help folding. And D refers to quaternary structure. So, and then E apparently is not correct. So the only correct answer is B. All right, so that's all for today.